Welcome. Today we are covering myelophthisic anemia, a distinct and clinically important form of bone marrow failure. Unlike anemias that arise from nutritional deficiencies or impaired cell production, myelophthisic anemia results from the physical replacement or destruction of the marrow space itself. When the marrow is occupied or obliterated by foreign or abnormal tissue, there is simply no room left for normal hematopoiesis to occur, and blood cell production fails across all lineages, red cells, white cells, and platelets alike. The most common and clinically significant underlying cause is infiltration of the bone marrow by a malignant neoplasm. This can occur with primary hematologic malignancies, but equally important is the scenario of metastatic solid tumors seeding the marrow from distant primary sites. Classic culprits include breast carcinoma, prostate carcinoma, and lung carcinoma, all of which carry a well-established propensity for skeletal and marrow metastasis. As tumor cells progressively colonize and occupy the marrow cavity, they crowd out and physically obliterate the normal hematopoietic tissue. The outcome is a global failure of blood cell production, not because the precursor cells themselves are inherently defective, but because the microenvironmental architecture that houses and sustains them has been destroyed and replaced. Less frequently, the same pathophysiological endpoint is reached through non-neoplastic mechanisms. Here, instead of tumor infiltration, the marrow space is destroyed by fibrotic replacement, a process in which normal hematopoietic cells are progressively supplanted by fibrous connective tissue. This is the prototypical non-neoplastic cause, and the condition is referred to as marrow fibrosis. The functional consequence is identical to tumor infiltration, a progressively shrinking hematopoietic compartment that is ultimately unable to sustain normal blood cell production. Turning now to the peripheral blood, myelophthisic anemia carries a critically important and diagnostically defining smear finding known as leukoerythroblastosis. This term describes the simultaneous appearance in peripheral blood of two populations of cells that under normal conditions are confined to the marrow and should never circulate. Their appearance signals severe marrow architectural disruption. The damaged marrow loses its ability to retain immature cells, and those cells spill prematurely into the bloodstream. The first of these two populations consists of nucleated red blood cells, also called normoblasts. These are immature erythroid precursors that have not yet undergone the normal process of nuclear extrusion that occurs before a red cell is released into circulation. Their presence in blood indicates that erythropoiesis is being driven under extreme stress and that the normal marrow barriers retaining immature forms have been physically disrupted. In the next video, we will discuss leukoerythroblastosis.